water quality trends in Southwest Florida. This is a presentation. By the way, this is going to be in 1 hour. There are going to be 2 presentations virtual, so hang in there. This presenter is Dr. Miles Medina, and he's already online. Thank you so much. And then before you go in, we have to mention that the purpose of this is to brief the TAC on a recent compilation of 20 years of water quality data to investigate the trends in CHNEP estuaries from 2000 to 2021, based on the monthly sampling throughout the CCHMN spatially stratified monitoring program, and the release of the water quality improvement actions 1.2 and 1.3, assessing and reporting the trends of the water quality, as well as assisting the consistent collection of the technical sounds along long-term water quality data. Thank you so much. It's all yours, Miles.
All right, hi,
You're
everybody.
free to speak.
Okay,
Yes.
Should I share the presentation on my end?
Okay, advance.
Yeah, okay.
You see it okay, Miles?
Oh, let me see. Yeah, there we go.
See?
Okay, cool.
Yes.
Yeah, perfect. All right, yeah, so hi everybody. So sorry I couldn't join you today in person, but it's a pleasure to speak with you again. So yeah, thanks for that introduction. I think with this group, you know, I think what I'm going to present today is not anything too surprising. I have presented to the TAC in the past and also the Water Quality Summit last year. But it is, I think, really important to get this kind of information on the record, published, and fortunately, this is work that's you know, started like something like 2 years ago, I believe, over 2 years ago, and we're finally in the final stages of review. So with any luck, this is going to get published in Estuaries and Coasts, hopefully before the end of this year. I want to start off by acknowledging the co-authors, including Jennifer and Nicole at CHNEP, Brandon at Charlotte County, as well as folks from other estuary programs in Southwest Florida, Eric Milbrandt at SCCF, and Chris Anastasio at the Southwest Florida Water Management District. And then also I want to acknowledge, of course, all the monitoring effort that's been gone on for the last couple of decades. This is really, this work was really, you know, inspired and enabled by this very consistent and monitoring effort that's produced a rich data set that really allows us to see what's been going on out there over the last couple of decades. So on the next slide, there's just a brief bit of context here. I don't think this group really needs to, I think this group's already familiar, but of course, the watershed for this part of the coast, for Charlotte Harbor, greater Charlotte Harbor estuaries, is very expansive. I think it covers something like 1/6 of the land area of the state of Florida. It includes that area in pink, which is of course the coastal drainage basins, basins for the Myakka, Peace, and Caloosahatchee rivers, as well as Lake Okeechobee and its watershed in green, which is connected via the Caloosahatchee River, connected to the coast. So this includes a lot of urban and residential development on the coast, as well as agriculture and mining further inland. Next slide. All right, so this CCHMN, or Coastal Charlotte Harbor Monitoring Network, was established about 20 years ago in the early 2000s, coordinated by CHNEP, with partners that include Sarasota County, Lee County, Swift-Mud, and others. And it's a very intentional sampling design for, you know, this very large open water area. The sampling design is such that we have 13 different segments or strata that are considered relatively homogeneous in terms of habitat and water quality. And each month, folks go out and take 5 grab samples from random locations within each stratum. So we get, in a typical month, 65 samples from throughout the system. And that randomization allows us to have some, you know, representative data for each stratum. We're not just collecting data from one particular point on the map, but it's sort of randomized within each one of these polygons. Now, a big part of the front end effort for this project was just sort of wrangling all of these data over the last 20 or so years. As you can imagine, with a bunch of different organizations cooperating, there's, you know, some inconsistencies in terms of how things are labeled and even which databases contain which data. A lot of the data are housed at FDEP's databases, Win and Storette. But of course, that's a process. It takes time. And so when you're looking in the more recent, the more recent period, sometimes you have to go into other databases, SwiftMud's database, CHNEP Water Atlas, and so on to sort of fill gaps. And sometimes those gaps exist throughout the record as well. So there was considerable effort here with a lot of help from Nicole and Brandon to sort of, you know, track down some of these records, make sure that we're only including data that are associated with the sampling design, not taking data that are collected in the area but not part of the sampling design, labeling things and just getting the data ready for this kind of long-term analysis. I don't have the link on the slide here, but if you're interested, the data set has been published. It's public. It's in a repository. So if you're interested as a researcher or anybody else in using the curated data set, just reach out to me. My email address is on the bottom of the slides, and we can share that. It's a very important output for this project. It's sort of the endpoint for the monitoring effort and then translation into research and so on. All right, next slide. Okay, so the monitoring program collects data on a couple dozen different water quality parameters. For this presentation and in the manuscript that'll be published, we focus on nutrients, nitrogen, and phosphorus, as well as chlorophyll. If you're interested in results for other parameters, again, email me and I can point you to some resources that we've got online that go into trends for some of those things. But here we're just kind of focused on nutrients and chlorophyll. Next slide. All right, so I briefly want to go over the methodology here. This is a kind of a different way of doing trend analysis. It's a bit more sophisticated, but it is a little more realistic than the traditional ways of doing trend analysis in terms of capturing nuances and complexity that we see over time in the data, as well as accounting for uncertainty. And this is a method that was published about 2.5 years ago by Marcus Beck and his co-authors. Marcus Beck, of course, works at the Tampa Bay Estuary Program. For this slide, I mean, these are results from the analysis. I'm not going to focus on, you know, what water quality parameter we're looking at or where. The idea is just to give a flavor for like how to read these graphs. This first graph shows the monthly data in gray over the 22-year period, 2000 through 2021. And the first step is to fit a GAM or a generalized additive model that has this kind of wiggliness that it can accommodate, this complexity it can accommodate to capture seasonality and also you know, changes over the long term. And so you see kind of with this blue curve sort of some sort of changes in direction over the long term, some ups and downs. It's not just a consistent, you know, up or down direction throughout the whole period. If we advance one more, we see the annual means that we can estimate from the GAM model, from the curve above. And so these are the annual mean concentrations for this constituent, this water quality parameter, with a confidence interval around it. And tracking the uncertainty throughout this analysis is really important because, of course, you might have a very volatile year. And so, you know, if you have a really high season, you have a storm, a major storm or something like this, you're going to have less confidence, a wider confidence interval around that mean, that estimated mean. Likewise, if you have a data gap, maybe in 2020 due to COVID, there were fewer samples taken in some cases. That can also translate into greater uncertainty around that mean. In the last 5 years of the record in this middle plot, you see a little gray line. So that's a 5-year trend line for those last 5 years, 2017 through 2021, and it's just flat. There's no up or down trend there. And one of the nice things with this analysis is you can do the analysis. We chose a 5-year window here, and you can slide that 5-year window across the entire period and estimate trends to see how that 5-year trend has changed over time. So what this final plot on the bottom shows is the slope value of the trend line over a sliding 5-year window. So that first red dot on the left corresponds to the slope of the trend line for the first 5 years. That's 2000 through 2004. Then we slide that over to the next 5-year window, 2001 to 2005. That is, we're sliding it over 1 year at a time. And we get a different slope. So the red dots here are statistically significant. They're above 0. So those are your uptrends. The blue ones are your significant downtrends. The white ones are, relatively flat. And so we see some changes in direction here. We see up, down again before sort of flattening out near the latter part of the record. And all the while, the uncertainty is being accounted for here. Okay. On the next slide, the final bit of the analysis involves tying the trend results, basically, are things going up or down, to some kind of absolute threshold, right? So the point here is that if you have an uptrend during a particular period, the conclusion is going to be very different if you're talking about an uptrend where concentrations are very low, and so it's not really a concern, versus an uptrend where concentrations are really elevated. And so that's, sort of an alarming, hotspot kind of situation. So what we did here was we just adopted thresholds based on the regulatory criteria, numeric criteria that come from Florida Administrative Code. These are estuary specific criteria, and the numbers are shown in the table there on the bottom. So for each trend, we can kind of classify it in 2 ways. Is it going up or down, and then where does it end up in that at the end of that 5-year period relative to the threshold? All right, so next we can get into the results. And I'm highlighting total nitrogen for 2 of the strata here on the slide. Of course, we have results like this for each of the strata. There are 13 of them. But we do see some things that sort of pop out, and some differences between the north and south that are interesting and illustrated here. So the first thing to note is that nitrogen concentrations have been elevated throughout the system, north and south. In the north, however, for example, the tidal piece segment, we see a consistent sort of uptrend during the last decade, 2010 to 2021, that ends up with conditions being elevated above the thresholds in each stratum. And you can see this, in each of these plots along the left. If you look at the sort of long-term tendency in the second half of the record in this top panel, you kind of see that along with the seasonality that's kind of oscillating each year, you see things kind of trending upward. You see that perhaps more clearly in the middle plot, where the annual means are sort of tracking upward. And then in the bottom plot, you see all these red dots. And remember that each red dot corresponds to the 5-year period that ends in the year where it's plotted. So for example, that first red dot is plotted above 2016. That corresponds to the 2012 to 2016 period. So what this means is that we're seeing, we've seen a consistent increase throughout the 2010 decade that ultimately ends up with nitrogen concentrations that are above the threshold, as indicated by the black squares along the top of the plot. Here, black is above threshold, gray is near threshold, and white is below threshold. In the south, for example, the Tidal-Caloosahatchee River on the right, things are a little bit different. The first of all, the numeric criteria for the Clusachi is lower. And so even though the concentrations aren't necessarily higher than in the tidal piece, with respect to the thresholds, the concentrations exceed the thresholds consistently throughout the period. So we see black squares along the entire period in the lower plot. And then the other interesting thing is that we see these ups and downs like we saw in the previous slide. So rather than seeing a consistent uptrend in the latter half of the record, we see this sort of these changes in direction over time that end up with a sort of relatively flat or stable kind of annual means more recently. I'm not sure what's causing, what explains this difference. It probably has something to do with the fact that the discharges from Caloosahatchee River are managed through flow control structures. And so maybe this reflects some kind of deliberate management of concentrations in the estuary. But it is interesting nonetheless. The point of this research isn't to necessarily explain what causes the trends, although there's other studies and things that we can use to help inform our interpretation of these results and offer some speculation as well. But at least this sets a sort of groundwork for what's the history of water quality, how has it changed over the last 20 years in this area. The next slide shows results for phosphorus. In general, across the system, phosphorus was low relative to the thresholds, and I think folks have different opinions about whether the thresholds in some segments are too high. We just took them for what they are. So we can quibble about whether the thresholds are appropriate or not, but at least relative to these thresholds across the system, with the exception of the Caloosahatchee River and Mount Lachey Pass, total phosphorus concentrations are pretty low and for the most part relatively stable. Finally, on the next slide, we have results for chlorophyll at 4 different strata. And here as well, we see that chlorophyll concentrations are pretty low, often trending downward throughout the system. And again, when I say low, this is relative to the numeric criteria. Now, the next slide shows a table sort of summarizing all the results across the system for different forms of nitrogen and phosphorus and chlorophyll. The way to read this is that the shape of each symbol indicates the trend direction. These are results for the final 5 years of the study period, 2017 through 21. So if you have an upward triangle or a downward triangle, that's an uptrend or a downtrend, respectively. A circle means that there was no trend during this 2017 to 2021 period. And then the color of the symbol indicates the category with respect to the threshold for the annual mean concentration in the final year, 2021. So when you see red, that means that the 2021 average was above the threshold, pink is near the threshold, and white is below the threshold. So, obviously what stands out in this plot is that nitrogen is elevated, especially across the southern segments that appear on the lower rows of the plot. Nitrogen is near the threshold in many of the northern segments. And throughout the system, this total nitrogen is dominated by organic nitrogen as opposed to inorganic. For phosphorus, we see things, as I said before, relative to the thresholds. Things in 2021 were pretty low, with the exception of the tidal Caloosahatchee and Mount Lachey Pass. And then finally, for chlorophyll, we see a lot of downtrends and concentrations that were low relative to the thresholds. So if you just consider these results, we see a lot of nitrogen, elevated conditions, increasing trends throughout the decade, which isn't the 2010 decade, which isn't necessarily shown on this plot. And yet chlorophyll concentrations don't seem to be responding to that. So what's going on? Where's all this nitrogen going? Well, we have to consider that there's a broader context here, right? This is just, looking at what's in the water column in terms of nutrients and phytoplankton, which is what we measure with chlorophyll. However, of course, on the next slide, we know that there have been massive seagrass losses since around 2016 or 2018. These are data that I got from Chris Anastasio on seagrass mapping. And of course, the seagrass losses coincide with landfall of Hurricane Irma in late 2017, and then the big 2018 bloom that lasted about 15 months, starting soon after Irma. So we're seeing, some response that's potentially connected to this eutrophication problem with nitrogen, some response in terms of seagrass. And also on the next slide, we see some of the transect data from macroalgae. This was provided by Mindy and Ariel at the aquatic preserves. So what these plots show is basically abundance of macroalgae at transects within each stratum. Each individual point is sort of a semi-transparent green color. So if you see a darker green, that means that there's a lot of points stacked on top of each other. And in certain subsegments, in particular, like the east wall on the left there, you see an apparent growth of macroalgae. And so it seems that, as has been hypothesized for a while now, that this nitrogen enrichment and eutrophication is manifesting more in proliferation of macroalgae, which is, of course, harder to sort of measure than chlorophyll, as opposed to manifesting as chlorophyll in the water column. So, I think in the research community, this is sort of recognized, the macroalgae problem is recognized not only in this part of Florida, but also throughout the world as a sort of major eutrophication pathway. But it's not necessarily reflected in regulations and the way that these systems are, and the way that the regulatory community, I think, approaches recognition of the eutrophication problem. So this broader context, this biological context, is very important. It's not just what do we measure in the water column, but also what are we seeing in terms of biological and ecological responses in the system. The next slide. We've got some takeaways here. So basically, the results for this analysis, probably nothing too surprising for this audience, but the results are consistent with the hypothesis that nitrogen enrichment and eutrophication is resulting in proliferation of macroalgae and loss of seagrass. Even though these results aren't necessarily that groundbreaking or resulting, I think it is important to get this on the scientific record, get it published, so that it can be referred to and serve as a foundation for management and policy intervention. Another important thing that is very clear from the trend analysis is that Hurricane Irma, you can clearly see the impact of Hurricane Irma. You see some jumps in nitrogen concentrations around 2017, for example. But those trends started well before Hurricane Irma. They were happening throughout the 2010 period before Hurricane Irma passed in 2017. And so what that suggests is that these increasing nitrogen concentrations create a sort of vulnerability that can make something like a storm like Hurricane Irma more impactful. And so I think going forward, as temperatures warm, we get more intense storms and so on, it really highlights eutrophication and nitrogen loading as an important thing that's happening all the time, that makes the system more vulnerable to those more extreme events. And then finally, I touched on some of the differences between the northern and southern segments that are not unexpected, because the northern part of the system receives discharges from the Mayaca and Peace River, whereas the southern segment is engineered or regulated flows from Lake Okeechobee and the Caloosahatchee and its basin. So you see different dynamics in the trends, north versus south. And then finally, on the next slide, just want to emphasize again, this methodology, I think, offers a lot of nice features beyond what we're used to seeing with typical trend analysis with the seasonal Kendall, for example, where rather than simplifying these complex dynamics to, you know, things trended up or trended down and that's it, we can take a look at the nuance, see how things change direction. You can look at different time frames. We looked over, you know, a sliding 5-year window, but you can look over a sliding 10-year window or whatever makes sense for the research. And you can also look at different metrics. You can look at the annual mean, like we did here. You can also look at the wet season maximum and things like that. And even though this is a more sophisticated and more sort of intensive analysis, it's easily scalable to generate results for as many water quality parameters as you like across as many locations as you like. So it's fairly automatic, which provides more time to, you know, think about what the results mean rather than just obtaining the results. Okay, so with that, the final slide has my contact info. Again, if you're interested in the curated data set or taking a deeper look at some of these results beyond nutrients and chlorophyll, feel free to reach out and I can share that with you. And if we've got some more time, I'd be happy to take some questions. Thanks.
Great. Thank you, Miles. Appreciate it. All right, we got time for 2 questions. 2 varying questions that might be on the public. Yes, sir.
Really interesting. You didn't see the expected relationship between nutrients and chlorophyll in the estuaries. Is there a way to see if there's a relationship between watershed nutrients and chlorophyll in the Gulf? Is that something you're also working on, or is that a possibility?
Yeah, great question. So, right, this kind of analysis just tells us sort of how water quality has changed. The next, you know, sort of question is what's driving it, right? And linking, discovering if there's a link between loadings from the watershed and what's going out, what's happening out in the coast. So yeah, I mean, you know, along those lines, a couple years ago, I published a paper linking Lake Okeechobee, Caloosahatchee discharges to red tide. I've got some preliminary results that seem to suggest a similar relationship for the Peace River. And I'm just starting a project that will trace conditions in the Peace estuary, sort of the tidal Peace segment, upstream into the watershed to understand, you know, which parts of the watershed are contributing most to eutrophication problems on the coast. So that's something I'm very much interested in. And something I'm going to be pursuing very, very soon.
Excellent. Thank you. Yes.
Yeah, real quick along those lines, since you're in here, James, is there an opportunity for Miles to connect with Rachel or some of the other folks that are actually doing all of that Peace River Basin analysis for the state so that we can all kind of
Yeah, Rachel Roach is a hydrogeologist just down the hall from me, and she's coordinating this group that's got overlapping goals and interests here. So I'll talk to Dr. Roach.
Okay.
So that's same sort of watershed scale, identifying the hot spots that are contributing to loading is a common goal there, and it makes sense that you should be communicating as much as possible.
If there's anything I can do to facilitate that, let me.
Right.
I wondered just really quickly, Nicole and Miles, if you could talk briefly about how you envision this potentially getting incorporated in the water atlas.
Sure. Yeah, I can share that we do have basically a task that will fund on the water atlas where, you know, there's already there is the trend analysis on the water atlas page, and this project on that Miles just kind of walked through. Actually, it kind of came out of a project where we were looking at expanding what trend analysis we have available through the water atlas, and maybe we're looking at a project in the next few months here to create either like a shiny dashboard or something like that to do additional parameters, and then share that through the water atlas trends page and have that available for you all because, you know, as Miles mentioned, this is data that everyone in this room has contributed to this enormous data set, and it's really prolific and a great way to capture what's been going on in our region for the last 20 years. So,
Okay.
Yeah.
Thank
And Miles, I don't know if you had anything else on that.
Mark.
Hey, Miles. You had a slide up there that showed there was basically no trend in nitrate and ammonia over the last 5-year period, which is when we've had the proliferation of the macroalgae and seagrass losses. Was there any trend for the 5 years prior to that in those parameters?
Yeah, that's a great question. So the issue there with the inorganics is they get consumed so quickly that they're often not detected in the grab sample. So for the manuscript, we're actually removing those trend results from there because what ends up happening is a lot of the data points are just reporting the detection limit. And sometimes the detection limits change over time. So it's easy to see a trend that's not really meaningful because we don't actually know what the concentration was because it was just too low to detect. So the sort of main conclusion across the board for things like ammonia and nitrate are just that, you know, it's so often undetected that it's not really appropriate to do a trend analysis. And that just goes to show how quickly the inorganic nitrogen is being consumed. So yeah, unfortunately, it's just sort of a limitation there, and what we can do just based on those detection limits.
Hey, well, thank you.